Hello everyone, welcome back to Python for Bioinformatics. In the previous lesson, we learned how to find where a pattern occurs using the find method. Today we are going to answer a different question. Instead of asking where something occurs, we'll learn how to determine how many times it occurs inside a DNA sequence. Now suppose we have this DNA sequence. We can already answer questions like whether A is present or where the first A occurs. But in biology, that's often not enough. We may want to know how many adenines are present, or how many guanines are present, or how many times a particular mutation or motif appears. So the real question today is, how do we count occurrences efficiently? Now let's try doing it manually first. Here is our DNA sequence, and I have marked each A that I see. And as we move through the sequence, we keep a running count. This works for a short sequence, but imagine doing this for thousands or millions of nucleotides. This quickly becomes impractical. So it would be useful if Python could perform this counting automatically. Before we continue, see if you can predict the total number of A's in this sequence. Python provides a very convenient method called count. The general syntax is sequence.count item. Here, sequence is the string we want to examine. The word count is the method name. And inside the parenthesis, we specify what we want to count. And Python then returns the number of times that item occurs in the sequence. Now let's use count on a real DNA sequence. In the first line, we store our DNA sequence in a variable named as DNA. And in the second line, we write dna.count_A. Let's read this from left to right. We start with the DNA sequence stored in DNA, then we call the count method, and inside the parenthesis we provide A, because A is the nucleotide we want to count. And we can see that the output is 8. The output is an integer, meaning a whole number. The number 8 tells us that there are 8 adenine nucleotides in this DNA fragment. So Python has answered our biological question automatically. The count method isn't limited to just A. We can count any nucleotide. And this time we use dna.count_G. The syntax is exactly the same, only the item inside the parenthesis changes. Python again scans the entire sequence and counts every G it encounters. The output is 4. This means guanine appears 4 times in the DNA sequence. Notice how easy it is to switch from one nucleotide to another. We don't need new logic or a new function. We simply change the value we want to count. Now here's something many beginners find interesting. The count method can count larger patterns too. Instead of counting a single nucleotide, we are now counting the pattern ATG. When Python sees dna.count_ATG, it searches through the sequence looking for occurrences of that exact pattern. And the output is 2. This means the sequence ATG appears 2 times in our DNA fragment. This is particularly useful in bioinformatics because we often care about motifs, codons, conserved patterns, and mutation signatures, rather than just individual nucleotides. Now let's connect these outputs back to biology. When we count A, G, T, or C, we are measuring the nucleotide frequency. And when we count a pattern like ATG, we are measuring the pattern frequency. And these counts help us understand sequence composition, identity motifs, examine codons, and analyze biological sequence more efficiently. So the count method is much more than a programming tool. It's a way to answer real biological questions. Now let's practice. Here's a new DNA sequence, and your task is to determine how many T nucleotides are present. Pause the video and try writing the code yourself before looking at the solution. Now let's check the answer. We store the sequence in DNA and then write dna.count T. Again, Python scans through the sequence and counts every T it finds. The output is 4. That means there are 4 thymine nucleotides in this sequence. And if your answer was 4, great job, you're already using count method correctly. Now let's summarize today's lesson. Our problem was finding how many times something occurs in a sequence. And Python's solution is the count method. The syntax is sequence.count item, and it returns the number of occurrences of that specified item. And importantly, the item can be a single nucleotide like A, or it can even be a pattern like ATG. Thank you for watching. Today we learned how to count nucleotides and sequence pattern using the count method. In the next lesson, we'll learn about the split method, which allows us to break a sequence into smaller pieces based on a chosen separator. See you in the next lesson.